he left Harvard and went on to do his master's and PhD at University of Michigan, where he worked with Daryl Kipke, doing all sorts of really interesting technology development. And Daryl Kipke, somewhere in the middle of that, called me and said, I cannot thank you enough for serving up my best PhD student. And I was like, look, I had nothing to do with that. I just got to get out of this guy's way. He went on then to do postdoctoral. Well, that was all very technology focused. And what's interesting, if you look at all of his papers and work, and he'll probably talk about this today, is in his postdoctoral work, he went on to apply this specifically in deep brain stimulation. And he really embraces the technology development, the modeling and computation, as well as the basic science of all this. So I think it's a really nice kind of package. He did his work at Cleveland Clinic and postdoctoral work with Cameron McIntyre. I just mentioned the three things he sort of focuses on. I just also want to mention he's one of the members of our external scientific advisory board for the McCamish program, and he's the perfect kind of person to help us really think about the basic science of diseases like Parkinson's disease, as well as the technology development, and help us take neurotechnology out of the laboratory into the actual setting where it can be applied. So final thing, Matt, you hopefully will get a sense that what I remember from 20 years ago is just this incredible enthusiasm and just like excitement about stuff. And I think as I've talked to him over the years following, he still has that. And so I think that's what I really remember about those days. So I'm going to turn it over to Matt and give you the microphone. And please, you're welcome. Yep.
Great. Thanks so much, Garrett. Can you hear me okay, everyone? Okay, great. Where I am today is really due to Garrett. I mean, he brought me into the lab. I had a sophomore class in physiological control systems. I don't know if you still teach a class like that, but it was bringing kind of linear control theory and applying it to physiology. And I saw that and I was hooked. I mean, it was the class itself, the demonstrations, and then Garrett was kind enough to bring me into his lab. I think I was the second person within his lab, and we were getting microelectrode recordings to work and dealing with a machine shop right next door that was emitting all sorts of noise. And it's just that early phase of just really excitement and passion, and I just very thankful to Garrett for bringing me in and helping me in my career. And so hopefully, you all have had those kind of moments where, there's been people in your career trajectory that have been just really great supporters, great encouragers, and hopefully some of you that are now in positions of like faculty can, pass it on to the next generation. So deeply appreciated to Garrett. I did have some pictures, but I didn't put those in. So if you want to see pictures, I can show them to you after the talk. So today what I wanted to do is talk about something near and dear to my heart is deep brain stimulation for Parkinson's disease, which kind of fits in with the McCamish Foundation and center that you have here. My grandmother had Parkinson's disease, and so that was one of the really big reasons that I was spurred on to try and develop treatments in this space. So what you're seeing here is just a little animation of one of our implants that we implanted into a non-human primate, and we can steer electric or direct electric current around this lead and have different kinds of effects, both on the neurophysiological level and also on the behavioral level. And so I hope to motivate some of that through this talk. So really the idea of interfacing technology is really gaining a lot of excitement in the field. And today I wanted to just talk about some of the work that the field has done and that we've done in this field of deep brain stimulation for Parkinson's disease. But some of the same principles and some of the same technology, can be adapted to, cortical stimulation, auditory stimulation, retinal stimulation. You've seen probably some of these devices on the market. So like the retinal stimulation with second sight. There's cranial nerve stimulation. We recently did a study with obstructive sleep apnea and targeting the hypoglossal nerve. And so there's a company, Inspire Medical, that has a treatment in that space. Vagus nerve stimulation, you've probably seen from the NIH, is really ramping up a lot of work in that space, not only for brain modulation, but also end organ function. Baroreceptor stimulation, phrenic nerve stimulation for central sleep apnea and spinal cord injury. Some really neat work that's coming out of spinal cord stimulation is treating paralysis. And it's this idea of placing electrodes below the lesion and stimulating and using sort of the reflex arcs that are still intact within the spinal cord to try and allow individuals to move. So we have some patients actually at the University of Minnesota that are 14 years out from their spinal cord injury and haven't been able to move their legs, and now they're able to actually move their legs. And what's fascinating is that at the end of the day, what's actually the most impactful is not been the movement, it's the autonomic control. And so by stimulation of the spinal cord, you're engaging sort of some of those sympathetic kind of reflexes and allowing patients, basic kind of like bowel function and orthostatic hypotension that they have difficulty with. Perineal foot drop, and then finally, a lot of work being done in the pelvic floor stimulation space. So I kind of want to kind of take you through just a little bit of a story in just one aspect of that, which is deep brain stimulation. So deep brain stimulation therapy is a technology that's implanting a lead of electrodes and applying a kind of isochronal stimulation pattern to a targeted brain region. And if we zoom in here, you can see this lead that was implanted into a patient that was targeting the subthalamic nucleus, or STN. So you're going to see a lot of that in the talk today. And so this area of the subthalamic nucleus is deeply embedded within the basal ganglia. And what I hope to kind of motivate through this talk is that there's not only the subthalamic nucleus, but there's a number of pathways that run along it, around it, and through it that are also implicated in this therapy. So what happens is when you apply stimulation, sort of before deep brain stimulation, here's an individual with Parkinson's disease, kind of akinetic movements, difficulty with gait, slowness, rigidity of limbs, some resting tremor. And then after stimulation, you can see sort of the outcomes, that the ability for motor control, or that individual to have some degree of motor control, is significantly enhanced. So this, oftentimes you might see a talk like, see examples of deep brain stimulation, you see these best case scenarios. But what I want to kind of let you know is that in the actual practical application of this, when we see patients in the hospital, there's a lot of variability in outcomes. And so here's a cross-center variability. You can see the distribution is skewed, and here's a within-center variability. This is a well-known DBS center, and you can see sort of the outcomes that are present. And so on the x-axis, you can see the improvement in what's called a UPDRS, or Unified Parkinson's Disease Rating Scale. And the part 3 of that scale is the motor component. And you can see the sort of the distribution. And really the idea is that we want to take this distribution and not only shift sort of the median, but we also want to tighten up that distribution. And so to do that, we really need to think about, where does this variability lie? Where do we see this variability? So there's certainly a number of different ways that variability happens. In one case, you can think of it as a neurosurgical targeting variability, that neurosurgeons are implanting these deep brain stimulation leads in a target that's less than a sort of a centimeter cubed. It's a pretty small target. And so you can see here's a case of targets of the subthalamic nucleus, that small little target, and where the leads ended up in a series of patients. There's also anatomical variability, that everyone's subthalamic nucleus is shaped, the morphology of it is shaped in a little bit of a different context. And there's also kind of functional anatomy variability. So within this target, you have regions that are involved in motor control, regions that are involved in associative processing, and regions that are involved in limbic processing. To give you an idea, if you were trying to do 2 tasks at once, maybe talk on the cell phone and drive your car at the same time, it would be difficult if you were stimulating maybe sort of the associative region of the subthalamic nucleus. The limbic area might be involved in more like mood processing. So if you're trying to control sort of the motor control elements of Parkinson's disease, really the target is that motor element that you can see there. Another factor that kind of it contributes to this variability is just patient-to-patient variability. This is a study that we did where we just took 12 subjects, and it was a part of a magnetoencephalography study, but we wanted to really characterize what are the symptomological kind of constitution of their actual motor deficits. And in this particular case, you can maybe just focus on maybe subject 1 to subject 2. They have the same level of this UPDRS disability kind of score, but you can see that the representation of what symptoms are actually present are very different. In subject 1, there's a high degree of rest tremor and kind of this postural kinetic tremor. And then in subject 2, there's a good deal of midline function. So this would be speech impairment, difficulty with gait, akinesia, kind of the difficulty making movements. And so you can have two different people. They have the same level overall of severity, but their actual kind of motor components are different. And so really, when we think about applying deep brain stimulation or other kind of therapies, we really want to focus on those symptoms in particular. So I've just listed some of the variability that you can see with patient anatomy, deep brain stimulation lead placement. Once your leads are actually implanted, then there's how do you design the electrode configurations, the electrical stimulation parameters to try and target, particular pathways, particular symptoms. These technologies, deep brain stimulation, are used in concert with medication. So oftentimes patients reduce their medication level, but the medication level, there's still medication in their system. And so how do you adjust stimulation to match sort of the medication level that they have within their system? And really all of this is geared towards trying to design ways to provide more personalized approaches to deep brain stimulation, kind of taking the patient into account. So what I want to do is I want to tell you kind of lead you through sort of a computational understanding of how these different pathways impact symptoms, talk about some algorithms, some machine learning algorithms that we've been using to try and automate that or optimize that process, and then getting into some of the fundamentals of like, what does deep brain stimulation actually do physiologically within the brain, and then kind of tying it back to the computational models at the end. So you may have seen some works of Svetlana Miocinovich, Dr. Miocinovich at Emory. We worked together in Cameron McIntyre's lab, and she's done some amazing things with computational models and recording electrophysiology data in patients recently at Emory. And she did one of the first computational models looking at subthalamic nucleus stimulation and trying to relate that to outcomes in non-human primates. And since then, this idea of taking computational models of predicting how electrical stimulation then impacts the biophysics of neuronal elements, cell bodies, dendrites, axons, relates to actual behavior is really blossomed and created a whole field unto itself. And so there's some really neat work being done in this space, and you can see kind of a range of different papers that have been coming out. And a lot of these are retrospective analyses. In other words, that patients are you see sort of clinical notes, you have the stimulation settings, maybe there's some imaging that you can kind of build into the developing these models. And what I want to show you is some of the things that we're doing looking at now prospective identification, where now we can take a model a priori and then use that to define which stimulation settings we think are going to be or provide the best outcomes for patients before they actually arrive in the clinic. So to get these models, you need some high field imaging data. So this is 7 Tesla imaging data that we've collected across multiple different sequences. And you take each one of those sequences and use those to create different types of models. So some of those might be models related to tissue voltages, tissue potentials, and other models might be related to the actual biophysics of individual pathways. And so you can kind of look at and develop these conductivity models, predict the inhomogeneity and the anisotropy of the actual tissue potentials, figure out where that lead of electrodes was located in a particular patient, build some tractography of these different pathways, and also segment the structures like the subthalamic nucleus. And then through this imaging, you can also identify within the subthalamic nucleus at these high fields the kind of a probabilistic map of the functional territories within this structure, within this small structure. So again, we're trying to target this motor area. So once you kind of have those pathway models, you can take and build those in around your deep brain stimulation lead and insert biophysical elements, so kind of like Hodgkin-Huxley dynamics, and really tune some of those to basic science recordings that have been done in those structures. And so these biophysical models are just built around differential equations, and you can solve those with a supercomputer. And so that's really what we do. And once you have that, then you can take and say, well, these particular pathway models, if I apply a certain stimulation parameter, how does that actually impact the ability of that particular pathway? Are we activating it? Are we generating action potentials? Are we suppressing kind of spike activity within a cell body? Are we antidromically activating that cell body? Are we, if you have dendritic connections, is there some kind of synaptic depression that's happening within that network or that synapse? And then you can relate some of those pathway activations to actual clinical outcomes. So here's an example of a study that we did looking at resting tremor. And you can see the clinical resting tremor and this subthalamic nucleus motor activation. So if you look at the associative or the limbic areas of the subthalamic nucleus, it doesn't relate, but in the sort of the STN, you see this sigmoidal curve. And it kind of like you need that sort of 30 to 40% activation in order to get a clinical resting tremor score from something high up to something that's more or less suppressed. So we have some driving questions with this, is that we can ask questions like, well, what pathways are associated with different types of motor scores? To what extent are those, to what extent do you need to activate a certain pathway to get an effect? Are there interactions with pathways? And I want to show you some examples of how you can have a therapeutic effect by stimulation in one pathway and an anti-therapeutic effect by stimulation in another pathway. And then ultimately, if we can identify these pathways, we can start thinking about hypothesis-driven DBS settings. And that's where I really think there's a lot of clinical need and a lot of excitement to get to, where these clinical trials that are being run for new forms of deep brain stimulation for a lot of like neuropsychiatric disorders, other forms of brain disorders, if you can go into a clinical trial with a hypothesis-driven way to apply stimulation, especially for stimulation that takes a while to kind of kick in, where there's the therapeutic latency is quite long, things like dystonia, depression, these hypothesis-driven ways might be pretty beneficial to having better outcomes with clinical trials. So one of the ways that we've thought about sort of building these models is you can take microscale data, and this would be like neural tract tracings. There's some really beautiful work that's been done in the basal ganglia looking at this. This is in primates. And then you can also take kind of macro scale, which is diffusion tractography. And if you've ever used diffusion tractography, you know that it can provide a lot of great kind of trajectory and tractography and pathways. But if you're trying to understand things within a nucleus, or if you're trying to understand tracks that are fairly small or that have like crisscrossing shapes to them, it becomes really difficult. And so one of the things that we've ventured into is this mesoscale kind of 3D view of anatomy. And so we're trying to build this up first in the non-human primate, and then we're kind of moving into humans, hopefully in the next year. And so the idea is we can, this is a collaboration with Tanner Aken at the University of Minnesota and my student Mary Penn. And so we kind of, we use this polarization-sensitive optical coherence tomography approach, where we can take a brain section and then provide serial kind of optical coherence scanning of that section as we slice through it. And one of the neat things about this approach is that myelin is a birefringent material, and so it depends upon the polarization of the light that you apply. And so from this, looking at sort of the birefringence of myelin, you can start getting really beautiful images of structures within the brain. And you can see sort of here's a snapshot of, if I use my cursor here. So you can look at the kind of the globus pallidus. Here's the external segment of the globus pallidus. Here's the internal segment of the globus pallidus. Two regions that are fundamental to the basal ganglia. You can see the part of the striatum called the putamen, involved in this part, kind of involved in more motor control. You can start seeing the thalamus and the subthalamic nucleus, and really start seeing these white matter tracks, these myelinated axons within the nucleus that we're actually trying to target. So here's two examples, and you can actually see this in thalamus as well. Hopefully I think you can see it on the screen pretty well. But here's a view of thalamus, and you can see these beautiful white matter tracks coming from the reticular nucleus, coming into thalamus, and you can see it's both sort of bottom up and top down coming in. And so when we're stimulating, right, the orientation of those axons probably matters, right, in terms of looking at the electric field. And you can see this is a cross-sectional view of capsule on the right. with thalamus. The capsule structure, there's fascicles within it. And the fascicular structure, I don't think really has been looked at much, because on the microscale, you get a single neuron, and the macro scale, it's just sort of diffusion tractography where it's more or less segmented. But there's just this beautifully rich data. And so right now we're just sort of in this data collection phase of collecting all this. But at some point, it's like, how do you create sort of models from this actual imaging data and connectivity models? And so I wanted to show you some videos of this. Hopefully this plays and you can see it. So this was just as a reference, the kind of the basal ganglia. You don't really see the subthalamic nucleus yet, but you can see the thalamus, internal capsule, caudate, putamen. Here's the external segment of GPE, and then the two kind of external and internal segments of this internal globus pallidus, which is very much involved in sort of the basal ganglia. So if I play this here, you can start seeing these rich fiber tracks, projecting through the basal ganglia. You can see now, like if you're looking in thalamus, some of these white matter fiber tracks coming through. And so when we're stimulating in these structures, the idea of having sort of a homogeneous structure is really it's not correct. It's that there's just this beautiful orientation of all sorts of different axons. And what you're seeing now with this approach is really this kind of, you can see sort of the two-dimensional piece to it as we're kind of going through, but also then the three-dimensional piece as these axons project into and out of the plane. Let me show you this one. This is the thalamus. So this is looking at it from a sagittal plane. So the first one on the left you can see is the coronal plane, kind of face on. We have a sagittal plane on the second, and we're looking at the thalamus right now at a lateral standpoint, or lateral, and then we're going to start moving medial. You can start seeing these fibers come in. There's the medial dorsal nucleus with the axons projecting, out to like prefrontal cortex. You can start seeing that some of the fibers, especially on the lateral side, coming up from the basal ganglia, substantia nigra, the globus pallidus, the deep cerebellar nuclei, probably elements of the cunate nucleus coming in. And so as we started building up this network of all this different axon trajectories, we started thinking about how electrical stimulation actually affects those. And one of the things to keep in mind when you're trying to apply current through an electrode and then use that to try and activate or generate an action potential with an axon, you really, if you have something tangential to your axon, you're really creating this iso potential across your axon, and that's not going to really drive current. You really want to think about this as kind of like the finite difference method that you probably have used maybe in the past for any kind of modeling, but it's really that idea of aligning your electric field to the axon is going to give you the greatest kind of bang for your buck in terms of activation. And so if you do this in a model, you can see that if you apply your electric field tangential to your axon, you're not going to get much activation, and if you apply it parallel to your axon, you're going to get sort of the greatest activation. And so theoretically, this happens, but what we tried to do, I'm getting low battery here. There we go. If we try and apply this actually experimentally, we looked at this in a rodent model, and did fMRI during stimulation. And so we had this 3-electrode configuration where we could vary the orientation of the electric field about the corpus callosum. And we looked at the kind of the barrel fields in S1 in this rodent, and then applied different orientations of that electric field. And what we saw was something very similar to our computational predictions, which was that when you align that field kind of tangential to those axons, you don't get a robust effect. And when you align it parallel to those axons, you do get a robust effect. And so we started looking at that kind of in the context of We did some pig work and looked at that with deep brain stimulation leads and saw the same things, that if we apply stimulation within thalamus and kind of align it to those axon trajectories, you see robust kind of fMRI in the pig, and you don't see as robust of activation when it's 90 degrees. If you've ever worked with pigs, we could talk offline, but it's the pig anatomy is very different than a lot of other animals. The motor cortex isn't really, you don't go sort of motor to sensory. It's kind of like more medial to lateral. We learned some things kind of through those experiments, so it's a pretty interesting model. So you can also apply this and think about sort of the electrodes, and if you applied monopolar stimulation, meaning that you apply current through one electrode in reference to some distant ground, more or less those field lines are going to have this kind of radial shape to them with most electrodes. If you use multi-contact monopolar stimulation, where you have two cathodes, now you can start looking at electric field lines that are tangential to your lead. And if you use bipolar stimulation, quite a bit of those field lines, at least between your electrodes, are now vertical or parallel to your leads. And so we've used this approach in some computational models in patients to try and align those to target a pathway called the hyperdirect pathway. So this is a pathway that projects from the motor cortex or aspects of the motor cortices to this area called the subthalamic nucleus. And using kind of an approach where you have kind of a multi-cathode configuration has a way to activate those axons more specifically. So then you get to the question of, okay, well, there's all these different pathways and they have all these different orientations. How do we actually make sense of trying to stimulate one pathway over another when they might have different locations, different angles to them? And that's where algorithms actually come in. So just to summarize kind of this first part, pathway activation models, it really provides a framework to associate neuromodulation with clinical outcomes. These axonal pathways through the basal ganglia, they have a broad range of orientations that are really absent in existing models. And you really want to align the electric field to generate those action potentials within axons. And just really there's a need for algorithms that can account for these multi-objectives that you might have to optimize your stimulation parameters. Because you probably, if you've ever done electrical stimulation or optogenetic stimulation, there's really a myriad number of different stimulation settings that you could use. And the question is, like, which ones would you actually use in your experiments? So there's a number of ways that you can think about algorithms. We've kind of looked at a couple of these. There's some classification, so you could do kind of machine learning, collect a whole bunch of data and try and classify it. You can frame the problem as a convex optimization problem. So if you think about kind of that electric field orientation, and if you align a certain, or if you have a certain pathway and maybe if you have another pathway that you don't want to stimulate, you could try and do some kind of like gradient descent, right, to try and find that global minimum. But one of the things that's challenging is that in the brain, with these electrodes, right, you could stimulate at this electrode or stimulate at this electrode, and in either case, you could activate a pathway. You're just activating it at different locations. And so it creates this non-convex problem that requires other approaches. And so one of the ways that you can do this is we've taken this particle swarm optimization approach. Has anyone ever come across particle swarm optimization? You have? Yeah. In what context? Oh, okay. Okay. So particle swarm optimization is one kind of evolutionary approach, and honeybees use this as a way to take sort of a collective problem and solve it in a way where each agent kind of goes out and tries to solve as best they can this problem, but they can also communicate. So honeybees can wag their tails, they can secrete pheromones, and you can converge on something that has multiple different local minimum to find something that's a global minimum. And so the analogy you could also take is you could say that a patient comes in, and maybe that patient is seen by 5 clinicians or maybe 100 clinicians, and each one is trying to figure out the best stimulation settings for that patient. And if each one was independent, you wouldn't necessarily get that global solution. But if each one of those clinicians talked to each other at each iteration, you'd be able to kind of form that global solution. So we really think of these particles and solving some objective function. So each particle has like an inertial velocity where it's going, It has a social velocity of where is the group dynamic happening, cognitive velocity of where it's found its best kind of effect, and then it creates a new velocity for that particle in the next iteration. And so just like honeybees, these particles kind of map this space, and then eventually they kind of swarm to find this global minimum. And one of the things that we started where we were thinking of this problem where we kind of assigned an objective function. We said, okay, pathway 1, pathway 2, pathway 3, and each one has a weight associated with it. But then we thought about, well, this is really, we could think of it more as a multi-objective problem, that there's probably multi-objectives that we might want. Maybe we do want to stimulate this pathway over this pathway, or maybe we don't. And so how could we define this kind of create this? And so we created this multi-objective approach, or use this multi-objective approach to really identify these Pareto fronts of the space. So it's kind of as these particles navigate, they're trying to push that Pareto front down to the kind of look at different multi-objectives at once. And so at the end of the day, it has this Pareto front, and each kind of point on that Pareto front defines a stimulation setting that you can use. And a lot of these are non-intuitive stimulation settings. So what you can get then is you have all these pathways. So we have a couple models that are now approaching like 15 different pathways, and you can see these Pareto spaces. And it's hard to visualize, so I'm just showing you three of the different different pathways. So internal capsule, the subthalamic nucleus, this hyperdirect pathway, the thalamic fasciculus, which is like the GPI projecting out to the thalamus and brainstem. And so you can pick one of these points in this kind of Pareto space, and then kind of identify the stimulation setting that you would use, and then apply that or see kind of what that would actually do in the patient. And so with some of these new tools for multi-channel stimulation, it works pretty well. And so we have a couple clinical trials now trying to test this out in patients, one for essential tremor and one for Parkinson's disease. So there's I think, there's a lot of different machine learning approaches that you could use, and I think there's a lot of excitement. I hope that, like, if you do some kind of stimulation perturbations within the brain, take a look at. There's a lot of papers that are kind of emerging in this space, not just our group, but a lot of different papers, and it's really an exciting way to try and reduce the dimensionality of your perturbation space in your experimental context. And thinking about sort of multi-objective approaches is really helpful. Okay, so the last part of the talk that I just wanted to show you is what happens in vivo when you electrically stimulate some of these areas, and what do you see sort of in the network. And I want to take you through some examples of stimulation in the subthalamic nucleus and sort of drive home some of the results from that. So we use a non-human primate model. It's a non-human primate model of Parkinson's disease. So it's a treatment with MPTP that induces the motor features of Parkinson's disease in our animal models. And we've worked with a number of vendors to develop these deep brain stimulation leads. So some of them are similar to what you see in the clinic, and some of them are ones that hopefully someday maybe get to the clinic. And they can have both kind of a banded electrode as well as split bands where you have electrodes around the actual lead. And so if you start stimulating in the subthalamic nucleus, what happens? Well, one of the ways that you can do that is you can try and look at spike activity between each stimulation pulse. So during the stimulation pulse, it's really difficult just due to the artifact to resolve what you're actually seeing, but kind of around it, kind of this interstimulus interval, you can detect spikes. And so we can do that, and we can look at single unit data. And if you look at what happens in the subthalamic nucleus during stimulation, one of the things that you see is that high-frequency stimulation inhibits the cell bodies. And the rationale for that is that, or the current kind of thinking is that you're really stimulating the afferents that are GABAergic going into the subthalamic nucleus, and that GABAergic innervation, that heavy GABAergic innervation, is really suppressing the cell bodies. But you also see that the pattern changes a little bit. So if you look at between each interstimulus interval, where does this inhibition kind of lie? It's right after the stimulation pulse. So within 3 milliseconds of that stimulation pulse, you see the cells that you see kind of a pronounced inhibitory effect. And after that 3 to 4 milliseconds, the cells more or less spike as they otherwise would, kind of without stimulation. So there's a phasic pattern to this effect. Now, if you stimulate in the subthalamic nucleus and now record downstream, where we have these glutamatergic projections to GPE and GPI, as well as probably stimulating some antidromic activation of those afferents, those inhibitory afferents, what happens? Well, it's kind of counterintuitive. You see this rapid increase in the spike rates of GPE and GPI cells. So if we're inhibiting the nucleus, why is it that we see excitation here? And one hypothesis could be it's antidromic activation. Another hypothesis could be that maybe we're somehow activating those axons projecting from the STN to the globus pallidus. So this was just a fundamental study from Jerry Vitek's group back in 2003 showing this. And what you see is that if you look at these interstimulus interval, these spikes tend to line up at certain phases of that interstimulus interval. And so you get sort of an early phase, kind of anadromic almost, and then you get sort of a 3 to 4 milliseconds, and then you get a more or less 7 milliseconds after the stimulation pulse. You see this kind of phase-locked spike activity. So we're changing the rates, we're changing the pattern of activity in this area, and this actually has a dynamic effect. So as you look over time, if here's your kind of, I've shown this as a peristimulus time histogram. So 0 is kind of one stimulation pulse, and then 7 milliseconds is another stimulation pulse. And you can kind of see the peaks that result are kind of excitatory effects that happen. And the dark gray line is the kind of off stimulation, and then the darker line is the kind of on stimulation effect. This actually changes over time, and it's actually over a period of 30 seconds that we see this kind of adaptation that happens in the ability of these cells to kind of keep up with that stimulation pulse. So initially it starts off where it's around 1 to 7, 1 to 8. So 8 stimulation pulses and you'll get a phase lock spike. And then over time it kind of adapts to kind of something in the more order of like 1 to 12. So it still keeps up with stimulation, but it just doesn't do it as faithfully. So there's some adaptation, and interestingly enough, this kind of like time constant seems to align with some of the changes that you see in the motor outcomes. What happens now in the nucleus of the motor cortex? So the subthalamic nucleus stimulation. We end at noon, right? Oh, yeah. I want to make sure there's questions. Okay. If you look at the hyperdirect pathway stimulation, so this is the pathway from the motor cortex to the subthalamic nucleus. What do we see? And we did, Luke Johnson and Jing Wang, we did this experiment where it was putting in this motor Utah array into motor cortex and then stimulating in the STN. And you see a range of different things. You see antidromic activation, you see excitation, you see suppression, and kind of this 32% at least of these cells were antidromically activated. So in other words, if I go back one slide, we're really antidromically activating that pathway, but additionally, we're activating pathways that then through the network result probably in some level of excitation or inhibition, or maybe it's just sort of a recurrent network within cortex where maybe one set of cells is antidromically activated that then has some kind of network modulation within those layers of cortex. So still trying to figure it out, but at least first order, there's definitely antidromic effects that happen. And again, these kind of change over time, where these cells like respond more robustly and then kind of like fade over time. And so you can see that here with the kind of time to antidromic peak and then the time relative to the start of stimulation. What happens during So one of the things that we've oftentimes wondered about is that there's changes in firing rate, changes in firing pattern, but these cells are encoding information. And so how does that information actually change? And so one of the things that we did was we started recording these cells, and this was early on. We just did a simple like general linearized model and just tried to relate sort of simple, if we're measuring rigidity, kind of that proprioceptive movement, how does that actually relate to some of the pattern firing dynamics spike dynamics that we were seeing in these basal ganglia regions. And so we did these experiments where we stimulated in the globus pallidus. It's a little bit bigger, so it's a little bit easier to do these kinds of experiments, and then recorded in the same structure. And what we saw was that if you stimulate at a subtherapeutic level, so it's like a lower frequency or a lower stimulation amplitude that didn't have like an effect on the actual rigidity, you really didn't see any change or much change at all of the response of that cell to that movement. But as you started bringing the frequency up or bringing the stimulation amplitude up, eventually you started getting these changes in how these cells were responding to those kinds of movements. And so this is a cell that kind of started having this strange kind of biphasic response to both shoulder extension and shoulder flexion. And we saw other cases where cells like completely lost how they were encoding that information. So here's a hip abduction adduction cell, elbow flexion and extension, and you can see sort of this noise level suppressed. And so we saw this like variegated kind of dynamics that were happening across these cells, and the number of cells that completely lost all information were pretty low, but the cells that had some level of information loss, be it position, velocity, acceleration, seemed to be present. So we're thinking of this as almost like not like a surgical lesion, but something more of like a leaky information lesion, where it's blocking some information through the network, but not all the information, so that this basal ganglia can still function in some way. And so if you look downstream of this in the thalamus, you can see that you still get sort of a leaky information effect, but it's not nearly as strong as we saw it at the site of stimulation. Okay. I think just for the sake of time, I wanted to show you a couple other things, but maybe I'll just skip to the end so we can have questions. Is that? Oh, we do. Oh, we do. Oh, okay. All right. Well, I'll show you this. This is some of our newer work. I'm really excited about it. A lot of the recordings that we've done in the basal ganglia, the thalamus and the cortex, and we started thinking, well, what's happening down in the brainstem? It's a really hard target. Alex on the left, on the left there. She was like audacious in trying to do this PhD project in selecting this one, because recording down in this structure is difficult, if any of you have done brainstem recordings. So she did this, and she recorded down in this area called the pedunculopontine nucleus. And the pedunculopontine nucleus is one of a few regions that are connected to this external globus pallidus. So the output of the basal ganglia goes into the thalamus, so the kind of the VLO, or the pallidal receiving area thalamus. It goes into the central median parafascicular nuclei of the thalamus, and it also goes down into the pedunculopontine nucleus, among a couple other regions. And this area is really interesting because of gait. And there's been some work done in the past where you can stimulate the pedunculopontine nucleus area, and you can cause, in like decerebrate cats, you can cause them to actually start walking. There's some really neat work coming out of Oli Kuhn's lab, or Sylvia Arber's lab, doing optogenetic stimulation of the different cell types within the pedunculopontine nucleus, and really starting dissecting how they relate to different aspects of gait. And so what we started to ask is, when we stimulate the subthalamic nucleus, are we actually modulating aspects of this? And we thought that maybe there's some differential modulation, because if you look at the axonal projections from the pedunculopontine nucleus to the basal ganglia, they actually project right through the subthalamic nucleus. And if you look at these kind of red projections, these inhibitory projections down to the subthalamic or down to the pedunculopontine nucleus, they project right around the subthalamic nucleus. And so we did this study in some non-human primates where we stimulated in the subthalamic nucleus and then recorded down in this pedunculopontine nucleus area. And we just were, curious to see, do we see that antidromic or orthodromic modulation? And so here's kind of, I'll show you a couple cells, and I'll show you the overall population response. So here's off stimulation, so kind of the cells, more or less spiking at 20 hertz. And then if you apply stimulation through that top contact, you see this, it's kind of like an inhibition, right, where these cells are inhibited kind of 2 to 3 milliseconds after the stimulation pulse. And if you stimulate down in kind of the central subthalamic nucleus, you get this antidromic response. And so it was really neat because we felt like we could, with those electrodes, we could start having differential effects on this brainstem structure. And so we could see that here, where actually what we saw is antidromic activation kind of in both areas, and it was that orthodromic inhibition was only on those top contacts. So then we started thinking about, well, could we translate this to humans? And so we built these models and tried to see if we could design stimulation settings, both targeting the subthalamic nucleus as well as that inhibitory projection on top of the subthalamic nucleus. And then we started building these pathway activation models, and you can see sort of that you could have this plot of a whole bunch of different stimulation, a whole bunch of different pathways, activation of the pedunculopontine nucleus to the globus pallidus, kind of the you could think of it as a ascending projection, and then the descending projection, which would be the globus pallidus to PPN. And then we modeled all these other pathways so you can kind of clamp on those or block on those so that you can maintain sort of the same level of activation across these other pathways, but really try and modulate just those two pathways, or the activation of those. And so we took 10 bilateral STN subjects and 20 deep brain stimulation leads and looked at sort of their step length. So in Parkinson's disease, one of the difficulties is stepping and kind of having that kind of a long step length. And so if you see sort of a negative change in step length, it indicates that it's actually worsening. And then a positive change in step length would mean that it's becoming more like a healthy control case. And what you can see is that so this was, in this particular study, this was without sort of designing the stimulation settings a priori. There's a range of different outcomes. And this kind of speaks to that variability that I talked about at the beginning of the talk. Naturally, when we're optimizing stimulation, it's optimizing for rigidity, tremor, bradykinesia, and really gait is one that's really difficult to optimize for. And so you see a range of outcomes. And so when we put this into a mixed effects model and looked at sort of the different pathway activations, blocking on those other pathways, you can see that this kind of PPN to GP has a kind of the confidence interval and the mean kind of are significantly elevated, kind of an increase in step length, and stimulating the GPI to PPN had an inhibitory effect. So we think going forward, sort of designing stimulation sequences that might be able to target these pathways might lead to better outcomes for gait. But on the other hand, and this is the last slide I have for you, the other hand, this same pathway, this kind of inhibitory pathway that had a kind of deleterious or just a negative effect on step size, right? That palatal to brainstem pathway that was inhibitory, it can actually be beneficial. So here's an example where we had a patient come in, and when we applied stimulation through contact E1, their tremor, resting tremor, was suppressed, but they developed these really bad dyskinesias. And we applied stimulation through E3, and their rest tremor wasn't improved, but they didn't have the dyskinesias. And so we tried alternating stimulation between E1, so they get the suppression of tremor, and then applying stimulation to that pathway down to brainstem or up to thalamus. And you could probably see in that video that the patient had a benefit where their tremor was suppressed and they didn't have those dyskinesias. So it kind of shows you that these pathways, it's not just sort of like, good and bad, right? That there's some nuance to how we might want to activate those pathways and in different contexts. So just to summarize here, high frequency stimulation induces rate modulation, pattern modulation. You really get that phase lock spike activity. The fidelity of that seems to adapt over time, and it'd be really interesting to track how that adapts over hours to days going forward. We think deep brain stimulation is inducing some kind of leaky information lesion, and I think it'll be really interesting to see how that translates through the network and how at some point we're getting motor control movements that seem more or less natural, and how does that happen? And there's definitely a lot of things that STN modulates regions outside the basal ganglia. I didn't get a chance to show it, but we have some data looking at the centromedian parafascicular nucleus, and STN-DBS definitely modulates that structure as well. So there's some really interesting work to be done. So just to summarize, if you kind of take-home messages, so axonal orientation heterogeneity with electrical stimulation is going to matter. Machine learning approaches are, I think, really neat tools that are helpful for building sort of these hypothesis-driven experiments that you can run. And I think in the future, where a lot of this is going to go is like symptom-specific modulation of thinking about particular patients, particular symptomological kind of presentations, and then how to identify which stimulation settings to use for those patients. So this takes a village to work through, and so there's some fantastic students that have come through, and this is their work that I presented. So Filippo and Annie and Allison and Alex and Mojgan, Edgar and Julia and Mary Penn, and then just some really outstanding collaborators, notably Jerry Vitek and Scott Cooper and Lauren Schrock and Tanner Aachen. So, thanks to the funding support, we're grateful for support from the NIH as part of a Udall Center, and Emory University, Georgia Tech has a Udall Center as well. And so it's a really neat way for our institutions to collaborate together. And so hopefully there's a lot more that we can do in the future in this space. And so the final two things I'll just plug is that we have this neurotech course. It's online. This has been my labor of love on weekends and nights. It's so for those of you that are interested in translation and commercialization, we developed a short course. It's free. It's online. And it's a whole bunch of videos with some kind of questions at the end of taking you through all of the different pathways or different ways that you could take some type of device and then translate it to the clinic and then also commercialize it. And then we also have a annual workshop that we're hosting on the 6th and 7th if you're interested. Come to Minnesota. It's nice, July 6th, 7th. Nice weather. And so we have people that we fly in from all over to do case exercises on translation and commercialization. And the last thing I'll just plug is that if you're interested in like a postdoc fellowship at all in kind of this translational space, we're just getting funded a T32 to kind of explore this in the context of brain disorders, brain cancer, autonomic dysfunction, and spinal cord injury. So it's kind of projects that would kind of embed you in the clinic to run experiments with clinical trials. So with that, just thanks for your attention. Thanks for the invite, Garrett. I really appreciate it. And yep, I'll take questions if you have any.
Okay, thanks very much. Yeah, I think there was something in there for everybody, from anatomy to technology.
Yeah,
cool physiology and patient.
If there's questions, I'll unmute this. Is that?
So, open for questions.
Please. Yeah, I think I found it.
And so maybe.
Oh, sure. Yeah.
So, my question is about the personalization, which I think is the opportunity. You talked about the variability at the beginning and the variability of placement and things like that. So do you see personalization would happen basically following with each subject? You would run some tests through the parameter space and then personalize? Or would you, before anything happens with the subject, you would say, oh, they're going to get this kind of stimulation?
Yeah, I think there's probably multiple stages to that personalization. So the question, in case the folks on over online aren't able to, could they hear it? Okay, great. So I think definitely it's kind of like we have to create tools that will allow for personalization to happen. So these deep brain stimulation lead technology has to adapt beyond sort of 4 electrode contacts to segmented electrodes, maybe having smaller electrodes that we could place actually multiple electrodes within this target structure to really align this electric field. But once the tools and technologies are there, then I think there's probably an element of this first programming where the personalization could happen. And then I think kind of like along the lines of your question, like probably later on, as people like go about their daily lives, there has to be sort of an adaptation responsive therapies. So maybe like one way to frame this would be is that there's sort of a personalization due to the anatomy and maybe some of the initial physiology. And then there's also probably a personalization related to like responsive therapies, where you're using sensor data, electrophysiology data, neurochemical data to do things sort of in real time or pseudo real time as patients go about their daily lives. Yep. Yeah, it's a really good
question over there.
Yeah. Yep.
Symptoms rather than pathway activation. How many different pathway activations would be able to reduce tremor in that one context? So I think you sort of offhand mentioned that you hit the motor region, that's the one that specifically seems to work. But I'm curious if that, there's sort of this cascade of degeneracies in DBS that kind of makes this problem very challenging to think about and sort of how you're grappling with that.
Yeah, I didn't hit the mic right ahead, but Jeff's question was looking at sort of the relationship between, or the kind of the disconnect between the, if I'm summarizing this, okay, the disconnect between sort of a simple kind of pathway activation to clinical outcomes, and then the complex physiology that happens in the context of what we've seen electrophysiologically. And there's a lot of complexity there at the electrophysiological level in terms of both the spatial piece as well as the temporal piece that I think we still don't have a really as robust a grasp as I wish we did. I think one of the things where when we start looking at the single cell level, we're seeing a lot of different things. I was talking to Eva earlier about this, and I think one of the things that we need to do is start building out like larger neural populations and recording those simultaneously to really understand what are things that are happening at the single cell level as well as the ensemble level, and try and map that to outcomes. And it could very well be that sort of this heterogeneity that we see might not necessarily be present on like an ensemble level in terms of encoding or something like that. What we've seen with the pathway activations is that there's certainly pathways that are very strongly related to outcomes, and then there's pathways that are weakly related, and so there might be some kind of multi-factor kind of presentation and kind of contributions to the actual therapy effect. But yeah, short answer, I don't know. Yep.
Do we want to ask if any of the online listeners would like to ask a question at this point?
There hasn't been anyone. No one's raised their hand.
Okay. All right. Well, so I have a question. I think Mike related a little bit to Annabelle's question earlier. When you started talking about the multi-objective sort of perspective and thinking about different ways that you want to engage a circuit or a network, I started thinking about parts of the space that you may be able to reach and other parts you may not be able to reach. And I just started wondering how do you approach that? If, for example, your objective involves stimulation in some way where it's almost mutually exclusive, like you can't get this and that at the same time. What happens?
Yeah. So it's become a real problem actually in the patients that we've seen, because what we'll end up having is we have this parietal front, and we're trying to do this. It's kind of like we're trying to create sort of a dot or trying to understand sort of like how we can create these pathway activations that are as far apart as possible. And in some of the patients, we just can't get there. And so what we have to do is it's kind of like a selectivity question of, we can do as best we can with a particular patient, but at some level, the technology that we have just won't allow us to kind of get that space. And so it's really these star patients that we have in the sense of their leads are kind of aligned in a way that we can really separate out these pathways. Then we can start asking more of these questions. And so it's a challenge that we've had to deal with. And I think as the segmented technology comes out more and more, and the multi-channel independent current controlled stimulation, comes out more and more, like the Boston Scientific device has that, I think we're going to be able to start creating those Pareto spaces that will allow us to separate out those pathways more. Yep.
There's an online question.
Oh, yeah.
So this is from Dieter. I'm going to unmute him.
Yes, hi, this wonderful talk. Hi, Matt.
Hi, Dieter.
Hi. I had a question also about the stimulation and the finding optimal parameters. Are you still looking for basically one stimulation setting for all pulses to get optimal outcomes, or are you within, say, 120 hertz stimulation, have like one pulse do one thing, one pulse do another thing, and then one pulse do another thing yet, so that you have basically 100 different combinations of pulses just for 1 second of stimulus? That would seem like a huge space, but at the same time, you could try and hit pathways differentially, even within the same stimuli.
Yeah, we have it. It's a really intriguing question. We've only been looking at electrode configuration in the sense of stimulation pulses that happen together. We've done a little bit with kind of alternating stimulation, but most of it is kind of based upon the idea that you kind of have this cumulative effect of stimulation across multiple electrodes simultaneously. There's some interesting work, kind of like your question. It's almost like you could create this onion of, different regions might be subjected to different types of stimulation pulses, and at different times. And the particular Pareto space and the multi-objective function that we've set up hasn't got there yet. But I think we have to do a lot more electrophysiology work just to try and understand what those different stimulation parameters do before kind of maybe venturing into the optimization problem. Yep.
Thanks.
Okay, well, so maybe we should at this point wrap it up. So let's thank Matt again.
Yeah, thank you, everybody.